Hey students, this is your Nandini ma'am. Today we are going to read chapter number 2, Cell Structure and Technologies from Focus Year 11 Revised. So let's get started. When you think about it, living organisms are amazing things. The secrets of their functioning have intrigued many generations of scientists over many hundreds of years. It has only been since the development of technology, such as the microscope, that the details of the structure and functioning of living things have been determined. This started with the discovery of the cell as the basic unit of all living things. Since then, it has been found that even though cells share some similarities, not all cells are the same. They differ in their structure, function, and organization. Some organisms consist of only one cell that has to perform all functions. Other organisms contain many different cells, each with a specific structure and function, organized to work together to ensure effective functioning. Types of cells. Even though there are many different types of cells in organisms, they can be classified as either prokaryotic or eukaryotic cells. Prokaryotic cells are primitive cells and have a much simpler structure than eukaryotic cells. There are many more prokaryotic cells on Earth than there are eukaryotic cells. Although these two types of cells have many differences, they share basic similarities, with each possessing a cell membrane, cytoplasm, and ribosomes, structures that produce proteins in the cell. Prokaryotic cells, with the word derived from the Greek pro, before, and kryon, nucleus, prokaryotic cells range in diameter from 0.1 to 5.0 micrometers. The four main structures that all prokaryotic cells possess are the cell membrane, cytoplasm, ribosomes, and genetic material. The cell membrane is responsible for controlling what goes into and out of the cell. There is no membrane surrounding the genetic material and therefore no nucleus. Most of the genetic material forms a large loop called the bacterial chromosome, with the rest in small circular rings called plasmids. The structures inside prokaryotic cells are not surrounded by membranes. They simply float in the fluid like cytoplasm, as does the genetic material. Structures that are present in some prokaryotic cells are a cell wall, villi, flagella, and a capsule. Villi are hair-like structures on the surface of some cells that allow them to adhere to nearby substances. Flagella are whip-like tails that provide the cell with locomotion. The cell wall protects the cell and provides structural support. The substances it is composed of differ depending on the type of cell. The capsule is a layer composed of complex carbohydrates outside the cell wall and protects the cell. Most organisms that are composed of prokaryotic cells are unicellular, a single cell. Some bacterial species clump together as a colony of prokaryotic cells that work together. Figure 2.2 shows a generalized three-dimensional diagram and a scientific two-dimensional line drawing of a typical prokaryotic cell. Prokaryotic organisms or prokaryotes can be divided into two main groups, bacteria and archaea. The cells in these groups are similar in both shape and size, but differ chemically in terms of their genetic material and proteins. Bacteria can be found in many different environments and can be either beneficial or harmful to living organisms. Archaea are unicellular organisms that are found in extreme and harsh environments such as hydrothermal vents and hot springs. Eukaryotic cells. Eukaryotic is derived from the Greek eu means true or proper and karyon means nucleus. These cells range in size from 10 to 100 micrometer and are much more complex than prokaryotic cells. Eukaryotic cells are characterized by a membrane-bound nucleus containing the genetic material of the cell. All of the internal structures of these cells are membrane-bound and are called as organelles. Each organelle has a specific function within the cell. Together, these organelles carry out all of the biochemical processes and reactions such as respiration, and photosynthesis that are required for the successful functioning of a living cell. Living organisms that contain eukaryotic cells are known as eukaryotes. These organisms can either be unicellular or multicellular. Paramecium, amoeba, and euglena are examples of unicellular eukaryotes. Multicellular plants and animals are composed of a variety of different types of eukaryotic cells. Key concepts. There are two types of cells, prokaryotic and eukaryotic. Prokaryotic cells exist as single cells and have no membrane-bound nucleus or organelles. Bacteria and archaea kingdoms contain prokaryotic cells. Eukaryotic cells have a membrane-bound nucleus and membrane-bound organelles. Investigation 2.1. A practical investigation to examine prokaryotic and eukaryotic cells. Living cells, whether prokaryotic or eukaryotic, possess structures that allow them to carry out the biochemical processes required for them to function effectively. These structures are organized differently in different types of cells. A variety of prokaryotic and eukaryotic cells can be examined using a light microscope. Only certain structures such as the cell membrane, nucleus, cytoplasm, and chloroplasts are large enough to be viewed using the microscopes available in school labs. For more details of such other structures in a cell, electron micrographs need to be used. Aim: to examine and compare a variety of prokaryotic and eukaryotic cells using a light microscope. Materials: light microscope slides, cover slips, onion, knife or scalpel, forceps or dissecting needles, eyedropper, optional. Methylene blue stain with eyedropper, paper toweling, 500 ml beaker of water for used microscope slides and cover slips, Embulia plant or an alternative freshwater oxygenating plant available from the local aquarium or biological supplier, plastic bag, disposable gloves, prepared slide of human cheek cells, prepared slide of bacteria, prepared slides of plant and animal tissues, for example, root hair, guard cells, frog blood cells, human blood cells, sperm cells, or neurons. Immersion oil. Risk assessment. What are the hazards? What risk does this hazard pose? And how can you safely manage this risk? Knife or scalpel. Sharp edges can cause cuts. Use knife or scalpel with care, hold by the handle, and keep fingers away from the sharp edge of knife. Microscope slides or cover slips. Sharp edges can cause injury if broken. Handle with care. Push gently on cover slip. Always focus by moving objective lens away from slide. Method. A plant cell onion. Remove the onion skin, cut through the onion and separate the layers. Carefully lift a thin section of onion tissue from the surface of one of the layers. Prepare a wet mount of this tissue using the technique outlined in the following steps. Cut a piece of onion tissue about 1 centimeter square in area and place this in a drop of methylene blue stain on the glass microscope slide. Be careful not to fold the tissue over on itself. Using a dissecting needle, lower the cover slip carefully to avoid the formation of air bubbles. Place a piece of paper towel over the cover slip and microscope slide to dry any excess stain. View under low power, then under high power. Draw a scientific diagram of 2 or 3 cells. Use a sharp pencil and draw single solid lines. Each diagram should be large enough, approximately 6 to 7 centimeter in size, to clearly show all structures visible inside the cells. Each diagram should have detailed and accurate labels. Label lines should be parallel if possible and should never cross each other. They should have no arrowheads and should touch the actual structure being labeled. Label the nucleus, cell membrane, and cytoplasm. Total magnification should also be recorded. B. Plant cell, Embulia, pond weed. Prepare a wet mount using a leaf from the tip of the plant. Place the leaf onto a drop of water and gently lower the cover slip. View under low power, then under high power. Note any streaming movement of the cytoplasm and chloroplasts. Draw a diagram of one cell. Label the cell wall, nucleus, chloroplasts, and cytoplasm. Record the total magnification. C. Enamel cell, cheek cells. Observe a prepared slide of stained cheek cells under low pressure, low power, and then high power. Draw a diagram of one of these cells and label the cell membrane, nucleus, cytoplasm, and any other structures you can identify. Record the total magnification. View prepared slides of other animal and plant tissues supplied by your teacher. Identify any different cell structures observed. Prokaryotic cell. Observe a prepared slide of bacterial cells. Use an oil immersion objective to give a higher magnification to observe the bacteria. Place a drop of oil on the cover slip above the specimen and center the oil immersion objective lens over the oil. Use the fine focus adjustment to obtain a clear image. Draw one bacterial cell and record the magnification. Note the size of a bacterial cell compared to a cheek cell. Results. Your results will include scientific diagrams including labels and total magnification of an onion cell, an embulia cell, a cheek cell, a bacterial cell, two other cells you observed. In a table, record the similarities and differences in size and structure between the cells you have observed and drawn in the previous question. Two plant cells, A and B, a plant cell and an animal cell, B and C, a plant cell and a bacterial cell, A and D. List the cells in order of size from smallest to largest. Discussion. Describe why methylene blue stain was used when preparing the wet mount of the onion cell. Compare the size of a bacterial cell to the plant and animal cells you have seen so far. Outline any cell details seen inside the bacterial cells. Identify two limitations of using a light microscope. Using your observations of the cells, suggest two differences between prokaryotic and eukaryotic cells. Describe how the prepared slides that you used compare to the ones that you prepared yourself. Describe how using oil immersion helped you to see the internal structure of the bacterial cell. Conclusion. Write a summary statement that relates to the aim of this investigation. A brief summary of what you did and what you found is required. Calculating total magnification. To calculate the total magnification when using a light microscope, the magnification of the ocular eyepiece lens is multiplied by the magnification of the objective lens being used. Total magnification is equal to magnification of ocular lens into magnification objective lens. You can use a light microscope to view cells. Total magnification is calculated by multiplying the magnification of the ocular lens by that of the objective lens. Technologies used to determine cell structure. Scientists in the 1500s used handheld magnifying glasses to view objects of interest. In the quest to see greater detail, scientists invented the first compound microscope. This microscope had two convex lenses placed at either end of a barrel and was the precursor to the light microscope. After much refining, this simple handheld device has transformed into the microscopes that are commonly used in the school laboratory. After further developments were made to the structure of these compound microscopes, such as placing them on a stand, adding focus knobs and a light source, scientists were able better to determine the structure of organisms. Robert Hooke in the 1660s was able to view and draw the structure and cork that led to his use of the term cella, leading to our term cell. Modern microscopes. Further refinements in the structure of compound microscopes continued over the succeeding years until we have those that are in use today. Light microscopes. In the compound microscope, such as those you use in the school lab, a light source passes through a condenser lens and then through the thin specimen. This beam of light then passes through the convex objective lens, where the image is magnified and viewed through the ocular lens. Compound light microscopes can produce images with a magnification up to 1500 into, depending on the lenses used. In microscopy, as well as magnification, resolution is also important in order to see the details of structures. Resolution is the ability to distinguish between two separate objects. It is the smallest distance between two objects where each can be observed as separate. For a compound light microscope, the maximum resolution is 200 nanometers. This means that the best light microscopes will only be able to distinguish two separate structures if the distance between them is 200 nanometers or more. If the distance between the objects is less than this, two distinct objects will be seen as one object. Both living and non-living specimens can be viewed using a compound light microscope. Fluorescence microscopes. The fluorescence microscope is similar to the light microscope. It has extra features that enable scientists to produce images of specific parts of cells. Structures beyond the limit of resolution of the light microscope can be seen using this microscope. The sample to be viewed is labeled with the fluorescent substance that will attach to the structures that the scientist wants to specifically observe. The sample is illuminated with a high-intensity source of light that causes the fluorescent substance to emit light. This fluorescent light is directed through filters that separate it from surrounding light, and the viewer is able to see only those areas of the sample that are fluorescent. Electron microscopes. Since the 1950s, study of microorganisms have been revolutionized by the development of the electron microscope. This instrument uses an electron beam instead of light and electromagnets instead of glass lenses. The interaction between the electrons and the object forms a viewable image on a screen. The use of electrons instead of light gives much greater magnification. Electron microscopes also have a much higher resolving power than light microscopes because electrons have a much shorter wavelength than light. The electron microscope reveals structure not only at the cellular level but also at the subcellular level. Materials that were formerly believed to have little or no structure have been shown to have elaborate internal organization. Many parts of cells were seen for the first time after the invention of the electron microscope. Other parts previously seen with the light microscope can be seen in far greater detail, providing increased knowledge of their internal structure. This has led to a greater understanding of their functions in cells. Features as small as 1/10 of a nanometer can be seen, including individual atoms. There are two main types of electron microscopes: the scanning electron microscope and the transmission electron microscope. The electron microscope shown in figures 2.10 and 2.11 is called a transmission electron microscope because the electrons are transmitted through the specimen. The TEM produces a two-dimensional image. It is the most common form of electron microscope. It can magnify up to 1,500,000 times and has a resolution of about 2 nanometer. A scanning electron microscope bombards solid specimens with a beam of electrons, which causes secondary electrons to be emitted from the surface layers of the specimen. The SEM has poorer resolution than a TEM, but gives excellent 3D images of the surfaces. Although there are many advantages in using an electron microscope, there are some disadvantages. For example, the specimen must be placed in a vacuum for viewing because air would interfere with the flow of electrons. As a result, living tissue cannot be viewed. The preparation of the specimen is very complicated and comes with the risk of introducing artifacts into the image. An artifact is something that is introduced into the image that wouldn't normally be there. Comparing samples prepared in different ways can identify whether artifacts are present. The size, expense, and the maintenance costs are much larger in this type of microscope than in others. Computer-enhanced technology to study cells. Digital processing of images produced by microscopes allows scientists to view cells in new ways. For example, cell scan software produces three-dimensional representations of cell structure, allowing a much greater understanding of cell structure and function. Our knowledge about cells has been helped by the development of advanced computer technologies to construct models that can be used to determine how molecules interact in cellular reactions. Confocal laser scanning microscopy can be used to highlight the three-dimensional structure of samples being studied. A laser produces a narrow, intense beam of light that is focused to a pinpoint on the sample, while all the surrounding, out-of-focus areas are not included in the image. This focusing occurs many times through the specimen at different levels. An image reconstruction program puts together the data from the images taken at different levels and constructs a three-dimensional image. These microscopes are often used to image the structural components of cells and have the advantage that the specimen remains intact. Key concepts. Early microscopes allowed scientists to see cells for the first time. Magnification indicates how much an image has been increased in size. The resolution indicates the minimum distance objects need to be apart to be able to be seen separately. Light microscopes pass light rays through thin specimens that are magnified by convex glass lenses. Magnification in light microscopes can be up to 2000 into maximum resolution 200 nanometer. Living and non-living specimens can be viewed using a light microscope. Fluorescence microscopy targets specific structures and uses labeling of these structures with fluorescent substances to see them. Electron microscopes use electron beams and electromagnets instead of light rays and lenses. Two types of electron microscopes are transmission electron microscopes and scanning electron microscopes. No living specimens can be viewed with an electron microscope. In a TEM, beams of electrons pass through a specimen and produce a two-dimensional image. Very high magnification and resolution are possible. In a SEM, electron beams bounce off surfaces and create a three-dimensional image. Confocal laser scanning microscopes use pinpoint laser beams to make images of many layers which are then combined into a three-dimensional model. Sizes of cells. Advances in biology over the last decades have pushed our view of the microscopic world to ever-decreasing sizes. Centimeters, millimeters, and even micrometers or microns are often too large to measure some objects. We are now in the nano age. Figure 2.14 demonstrates the size of the objects seen by the naked eye, a light microscope, and electron microscope. Drawing scaled diagrams. When drawing different types of cells, such as those viewed in Investigation 2.1, it is useful to draw these cells to scale. This allows a more accurate representation of the size of these cells. Diagrams that are drawn to scale will always contain a scale bar, which indicates the scale to which the diagram is drawn. The scale bar represents a specific whole number of micrometer and is often 1 to 5 centimeter in length. In order to draw the scale bar, determine the actual length of the object to be drawn. When the actual length of the object determined above is divided by the length of the proposed drawing, a whole number should be obtained. For example, length of cell is equal to 8 micrometers and length of drawing is 4 centimeter. So actual length over length of drawing will be 8 micrometer over 4 centimeter, which means scale will be equal to 2 micrometers over 1 centimeter. Therefore, a scale bar 1 centimeter in length would represent 2 micrometers, or a scale bar 2 centimeter in length would represent 4 micrometers, and so on. Okay, key concepts. 1 centimeter is equal to 10 mm and 1 mm is equal to 1000 micrometer. 1 micrometer is equal to 1000 nanometer. Scaled diagrams must always contain a scale bar. The scale is used to can be calculated by dividing the actual size of the cell by the length of the diagram to be drawn. Organelles in cells. All eukaryotic cells contain membrane-bound internal structures called organelles, each with a specific structure and function. Different organelles share the common feature of having internal structures that are enclosed by their own membrane. The membrane may be either double or single in structure. Although each organelle has a different structure and function, they all work together to contribute to the effective functioning of a cell as a unit. Many organelles have maximized the amount of surface area they have available to carry out their particular functions. One of the differences between organelles in their size. Some organelles, such as the nucleus, vacuoles, and chloroplasts, can be seen with a light microscope. Details of other organelles, such as mitochondria and ribosomes, can be seen only by using an electron microscope. See Table 2.1. The arrangement of organelles also varies depending on whether the cell is a plant or animal cell. A generalized diagram and line diagram of a eukaryotic animal cell is shown in figure 2.19 and a plant cell in figure 2.20. Plant and animal cells contain many of the same organelles. The plant cell, however, possesses a cell wall, chloroplasts, and a large vacuole that the animal cell does not. Figure 2.21 summarizes the organelles that are found in both plant and animal cells. It also shows the organelles specific to plant cells and those that are only present in animal cells. Membranes, selective boundaries. The cell membrane, alternative names are plasma membrane, cytoplasmic membrane, and plasmalemma, surrounds the cell contents in all cells and separates the cell contents from their surroundings. It controls the passage of water and other chemical substances into and out of cells. It is a selective barrier permitting the passage of only certain molecules into or out of cells. This property gives the cell membrane the feature of being selectively permeable. Both plant and animal cells have a cell membrane. The membranes surrounding organelles are also selective in allowing only certain substances to pass between the cytoplasm and the organelle. Protoplasm. It is in the protoplasm of cells that the functions essential to life, such as making cellular products and respiration, are carried out. The protoplasm is the living content of a cell that is surrounded by the cell membrane. The protoplasm is composed of the nucleus and the cytoplasm. The cytoplasm, that part of the protoplasm outside the nucleus, consists of a liquid called the cytosol in which there are dissolved chemical substances, for example, ions such as chloride ions, suspended organelles, and insoluble granules. Approximately 90% of the cytoplasm is water, the medium in which all cells chemicals are dissolved or suspended. Nucleus, the control and information center. The nucleus appears as a large spherical or oval structure in the cytoplasm. It is colorless, transparent, and slightly more jelly-like than rest of the cell. Most organisms have one nucleus per cell. The nucleus stores the information needed to control all cell activities. It is therefore essential for the nucleus to be able to communicate with the surrounding cytoplasm. Electron micrographs reveal that the nucleus is surrounded by a double nuclear membrane or nuclear envelope pierced by tiny pores. These pores regulate the passage of substances between the nucleus and cytoplasm, allowing communication between them. The nucleoplasm or the nuclear sap is the liquid part of the nucleus in which the chromatin material is found. Chromatin is made up of protein and nucleic acid. The deoxyribonucleic acid, DNA, is stored in the nucleus. It is a very large chemical that holds in a coded form all the genetic information, the blueprint necessary to control the cell's function. This DNA contains the hereditary information of an organism that gets passed from one generation to the next. Within one organism, the information stored in the DNA of each cell is the same. Before a cell divides, the information on the DNA must be copied so that it can be transmitted to a newly formed cell. The chromatin separates into short, thick, separate, rod-shaped structures called chromosomes, which become visible in dividing cells. So named because chromosomes take up stains easily when being prepared for microscopy. Each species of organisms has its own particular number of chromosomes. For example, humans have 46 chromosomes, a platypus has 52, a lettuce has 18, and a camel has 70. The nucleolus is a dense, granular region commonly seen within the nucleoplasm. It contains a large amount of nucleic acid, some DNA, but mostly RNA. The nucleolus is responsible for the manufacture of organelles called ribosomes, the essential machinery of the cell. Endoplasmic reticulum, transport and processing of proteins and lipids. The outer nuclear membrane is usually continuous with a network of flattened, interconnected membranes, the endoplasmic reticulum. The ER provides a connection of pathways between the nucleus and the cell's environment, allowing intracellular transport. The immense folding of the sheets of membrane increases its surface area. ER may have ribosomes attached or may have no ribosomes. The main function of ER is transport, but it also plays a role in processing cell products. Rough ER folds and processes proteins made by the cell, and it can also synthesize lipids. Smooth ER is the main site of lipid production, essential for membrane repair and manufacture. ER may also transport substances from one cell to another in plant cells, passing through channels called cell pits in the cell wall. Ribosomes, protein synthesis. Ribosomes are small organelles that appear as dense, rounded granules in electron micrographs of cells. Their small size increases their surface area. Each is made of the chemicals RNA and protein, and they are the machinery that carries out the genetically coded instructions of DNA to produce any proteins necessary for cell functioning and structure. Amino acids are joined in order to form polypeptides, the structural unit of proteins. Ribosomes may be found free in the cytoplasm or scattered over the surface of ER. Newly synthesized proteins pass from the ribosomes into the ER where the protein is folded. Golgi bodies, packaging and sorting the substances. The Golgi body, also known as Golgi apparatus, is also made up of flat membranes, but it differs from ER in that it does not have ribosomes attached and its membranes are arranged in stacks of 4 to 10. The Golgi body is easily recognizable by its curved shape on one surface where vesicles can be seen. Budding off. This surface is called the forming phase, and the vesicles are evidence of the secretory function of Golgi bodies. The opposite surface may be convex or flat. Golgi bodies process, package, and sort cell products. They're involved in adding proteins and carbohydrates to cell products, and they also provide a membrane around the cell products to package them. The membranes provided by Golgi bodies vary and serve as a packaging label. Features of the membrane are used to sort these products, determining where they will end up. They may be transported within the cell to wherever they are required, or they may be secreted from the cell. Lysosomes, digestion and destruction. Organelles within the cytoplasm of cells will eventually reach their use-by date and wear out. Instead of wasting the raw material that make up these organelles, the cell has a clever method of recycling and reuse. This is the job carried out by lysosomes, special organelles found within the cytoplasm of animal cells. Lysosomes are formed by the Golgi body. They contain digestive enzymes that are responsible for splitting complex chemical compounds into simpler ones, such as when proteins are broken down into amino acids. These simpler molecules can then be used as building blocks for new compounds and organelles. Sometimes, lysosomes may destroy the entire cell. This process is known as apoptosis or programmed cell death. It is a deliberate action by the cell to destroy old and damaged cells. Under the direction of the cell, the lysosome membrane ruptures, releases the enzymes, which then digest the contents of the cell, killing it in the process. Mitochondria, cellular respiration. Mitochondria are the powerhouses of a cell, producing energy in the form of energy-rich molecules by the process of cellular respiration. Mitochondria are usually rod-shaped but may be round, that they vary in both shape and size. Mitochondria are smaller than the nucleus and chloroplast but larger than ribosomes. The number of mitochondria in a cell depends on how much energy the cell needs to carry out its functions. A less active cells contain few mitochondria, whereas very active cells have hundreds or even thousands of mitochondria. For example, active liver cells contain 1,000 to 2,000 mitochondria. Just as machines in a factory need electrical energy to work, so cells need energy in the form of a chemical called ATP to work. Adenosine triphosphate. Mitochondria combine oxygen with sugars during the process of cellular respiration to release energy in a form ATP that the cell can use. A double membrane surrounds each mitochondrion. The outer membrane gives the mitochondria their shape and allows the passage of small substances into and out of mitochondria. The inner membrane is folded into fine finger-like ridges or cristae, increasing the surface area for attachment of groups of enzymes responsible for producing energy for the cell. These appear as knob-like materials on the inside of the cristae. The central space in a mitochondrion is filled with fluid and is termed as matrix. It contains mitochondrial DNA and enzymes that give mitochondria the unusual feature of being able to replicate themselves. The mitochondria divide by pinching off and then growing, something that usually occurs in very active cells or cells that are able to divide. Vacuoles, storage, and support. Vacuoles are large, permanent, fluid-filled sacs in the cytoplasm of mature plant cells. Each vacuole consists of a watery solution called cell sap surrounded by a single membrane, the tonoplast. Cell sap contains substances such as mineral salts, sugars, and amino acids dissolved in water. It may also contain dissolved pigments that give color to the cells. For example, reds, pinks, and purples seen in some flower petals. Besides having a storage function, vacuoles play a very important role in providing support to the plant cells. By filling up with water, the vacuole pushes outwards with the cytoplasm, exerting a pressure on the cell wall, keeping it firm. As a result of the outward pressure of the cell contents and the resistance of the cell wall, the cell becomes firm or turgid. Small temporary vesicles may sometimes be found in animal cells, but these do not play a role in cell support or so permanent vacuoles that give turgidity are considered to be a feature exclusive to the plant cells. Chloroplasts, photosynthesis. Chloroplasts are the organelles that are green in color due to the presence of a pigment called chlorophyll. Chloroplasts are responsible for photosynthesis, the manufacturing of sugar in plants, using the energy of sunlight. Chloroplasts are not present in all plant cells. They are found only in the green tissue of plants that can photosynthesize. Under the light microscope, they appear as green, disc-shaped structures smaller than the nucleus. An electron microscope is needed to see the detailed interior. Chloroplasts belong to a group of organelles called plastids, which are biconvex in shape. Plastids contain either pigment or nutrients and vary in color. Chloroplasts are green plastids. They are larger than mitochondria, but are similar in that they also contain their own DNA, and the number of chloroplasts per cell varies. A double membrane surrounds chloroplast. This allows substances to pass between the cytoplasm and the chloroplast. Unlike mitochondria, the inner membrane of the chloroplast is not folded. The liquid part of the chloroplast is called the stroma, and it is here that the stacks of membranes called thylakoids are found. Each stack or group of thylakoids is termed as a granum, and the chlorophyll is found on these membranes. The layering of the membranes increases the surface area over which chlorophyll occurs, allowing a large amount of sunlight to be absorbed by the process of photosynthesis. This captured energy of sunlight is then used by the plant to make simple sugars. All the enzymes needed for photosynthesis are present in the stroma, and simple sugars made during photosynthesis are stored in the stroma as starch grains. Plant cell wall, shape, and support. The cellulose cell wall that surrounds plant cells differ from the cell membrane inside it. The cell wall is not selective in terms of what substances it does or does not allow into the cell. Its structure allows it to provide strength and support. The strands of cellulose fibers have a little elasticity, thickened, and they are somewhat flexible so that they can resist pressure. Some cell walls are thickened with additional chemicals that make the walls hard and woody, for example, in tree trunks, or that provide waterproofing. Cell walls are permeable to most molecules. Centrioles, spindle production in cell division. A dense granular structure, the centrosome, is often found near the nucleus in animal cell. It consists of two centrioles which play an important role in cell division, forming the spindle which holds chromosomes in a dividing cell. Cytoskeleton keeps organelles in place. Organelles are not randomly scattered within a cell. Their distribution is organized and they are held in place by a network of tiny microtubules, microfilaments, and intermediate filaments called the cytoskeleton that extends throughout the cytoplasm. It is the framework for the shape of the cell movement, organelle movement, and cell division. Key concepts. Organelles are membrane-bound internal structures, each with a specific function to ensure the efficient functioning of the cell. Most organelles have structures that maximize the surface area. Organelles and cell structures vary in size, and with only the cell membrane, cell wall, nucleus, chloroplast, and vacuoles being big enough to be observed using a light microscope. The function of organelles and other cell structures is summarized in Table 2.2. Membranes. Selectively permeable boundaries, control the movement of substances into and out of the cell or organelle. Protoplasm, the living content of a cell that is surrounded by the cell membrane. Nucleus, the control and information center. Endoplasmic reticulum, transport and processing of proteins and lipids. Golgi bodies, packaging and sorting the products. Ribosomes, protein synthesis. Lysosomes, digestion and cell destruction. Mitochondria, cellular respiration, production and storage of energy. Vacuoles, storage and support. Chloroplasts, photosynthesis, plant cell wall, shape and support. Centrioles, spindle formation or production in cell division. Cytoskeleton, cell shape, organelle placement and movement and cell division. So, in this video, I'm going to record till 2.4. To hear the next part, just wait for the next video. Thank you and happy reading.